Yeah, good morning to all, and welcome to the Abhignya Biotechnology Consultant Bioinformatics workshop. So for the module 1, so today we are going to learn the basics of bioinformatics. So before going to the module, I would like to give a brief introduction about Abhignya Biotechnology Consultant and when it is established and what is our motto and what is the reason behind to start the consultant activities in the field of life sciences. So during pandemic, I realized that the students are facing so many problems in the field of life sciences, especially they're not going to the labs. They're not learning the laboratory experiments, and they're not updating the skills, which is essential to enter into the industry level. So to keep on in mind, while interacting with all the biotech industry entrepreneurs and research scientists, they advised me to establish a center which can help for the beneficiary of the students. So we are trying to fill the gap between the industry and academia and research centers which are established across the nation. The reason behind why to start our services in Hyderabad is, if you see the geographical, Hyderabad is the world's topmost vaccine producing city. And we have the Asia's largest genome cluster in Hyderabad. And very soon we are going to have the Asia's biggest pharma city, which is going to start in a couple of months in Hyderabad. And if you see the national institutions, we are having IITs, NITs, Central University, and there are more than 9 CSIR labs are located in Hyderabad. So by keeping all this, we are having a plenty of job opportunities in these industries as well as research academic institutions. So to enter in this field, you must learn something which is required for the industry as well as the research institutions. So by keeping this in mind, we have started the Abhignya Biotechnology Consultant as a skill development center in the field of life sciences, established under Ministry of Small and Medium Enterprises, Government of India. So to create the opportunity for the youngsters, the students and the faculty and the research scholars, we started this Abhignya Biotechnology Consultant because we know that how the industry is growing and the industry is having the vast opportunities and filled with plenty of opportunities for the students. So the major sectors, if you see in the area of life sciences or biotechnology, they are research and development, regulatory affairs, quality control, and clinical research. So they are the more major areas where we can find employment for our career. So if you see, when I visited the industry, while interacting with the entrepreneurs, they are searching for the young minds who is having the strong academic knowledge with experimental, innovative, and critical thinking to work individually and in team. So this is the area where you can build a strong network group and strong foundation for your future without doubt. The vision of Abhignya Biotech Consultant is the quality of academia with experimental knowledge, career development, and dynamic future in the biotechnology or life sciences. Our mission is to create an opportunity for all students, especially young minds, never before, like by enhancing the student skills as per the industry requirements with strong academic knowledge, and to promote them to become an entrepreneur in the field of life sciences as well as the biotechnology. So we have started in the year November 2020. So successfully completed 1 year, and we have organized a plenty of webinars in collaboration with the various institutions located in Hyderabad. And we are happy to say that we are working as a knowledge partner for 2 prestigious institutions in Hyderabad. One is Government City College, a 100 years old city college, and Nizam College, as well as the St. Francis College and St. Mary College. So the services are the internship programs, the science project dissertation work, which is mandatory for your to complete your graduation and post-graduation. And we are organizing workshops and skill development programs, which will help the students to enter into the industry as well as the research institutions. And I said that already we are working as a knowledge partner for academic institutions, and we are making them to enter in the industry. So to see what is exactly industry doing and what is how industry is functioning, and the responsibilities of industries and the responsibility of the individuals who are entering the industry. And the special service of our Abhignya Biotechnology consultant is we support the startups and innovations and providing the incubation services. Because if the students is having the idea which is having some commercial value, so we will validate that idea behalf of you, and we'll make you to interact with the investors and make it possible to reach your goal, and the product will be released in the market by the investor itself only. So we'll support you in that area also. And the most important thing is the career guidance and employability. So we will definitely guide you through their career from A to Z. At the same time, we'll try to create some employability with collaboration of the various industries located in Hyderabad, especially. So I want to give you a small slide presentation from our side. When you choose your career in the field of life sciences or biotechnology or any science, the things that you should not do, first and most important thing is do not compare yourself with others, because we know that our DNA is unique. Do not blindly trust your data. When you produce the data, especially when you're doing the experiments, when you get the data, don't think that you got the results. There might be a wrong results you get. And do not suffer alone. Suppose if you are facing any problem in the science, especially by reading or understanding the concept or understanding the experiment and understanding the procedure, do not suffer alone, and interact with your friends or interact with your mentor or interact with your teacher and discuss with them and see the possible way to get out of that suffer. And do not believe that more work is always better, because sometimes the hard work also pays, at the same time smart work also pays you better. So don't believe that more work is always better. Don't sit in the ideal in the classrooms. Don't sit idle in the laboratory for longer duration, and you say that I am working so many hours, I am working from morning to evening, but I'm not getting results. So do not believe that more work is always better, and sometimes you need the rest also, and take the best possible way that how come you can settle this one. And do not grow your records organically, okay, and maintain your records that what your experiments you are doing in the laboratory, and maintain the records that what you're learning, and everything is important. Do not get stuck after one failure. This is the most important things, because when you fail in one process, the failure will make you to understand the obstacles and make you understand the why did you get failed, and you'll get most experience to resolve the problems. So this is what I want to say you when you choose a career in the field of science. So we want to say that convert the desire that what you are having the desire into a physical reality, and we'll make it for you be of Abhignya Biotechnology consultants. Okay, do not go to your grave with your best work or idea inside of you. Choose to die empty, because there is no use if you're having your idea in your brain if you're not validate and make it to a physical reality. So the famous quotation is, use your right brain to become intelligent, to be intuitive, and to lead and succeed in life. That means your left hand should use frequently to reach your goals. And thank you. This is our address, and we'll provide you the what regarding your career, regarding your future in the field of life sciences. We are always at your doorstep. Just send us an email or text us on WhatsApp, and we'll respond to you 24/7. Thank you so much for listening. So with that, I conclude that the brief introduction of Abhignya Biotechnology consultant. Now it's time to introduce for today's resource person, Dr. Chikoo, and she has completed her BTech and Masters and PhD in the field of bioinformatics. And she has visited the Harvard Medical School on the student exchange program in the year 2021. And presently she is working as a research scientist at 3B Limited, which is situated in Bangalore. And she has published more than 10 research papers in the reputed journals. And she has experience in the field of computational biology, chemoinformatics, proteomics, genomics, NGS, and so many the bioinformatic tools. And she is having a tremendous knowledge and experience in the field of bioinformatics. So with that, I would like to, and thank you, Dr. Chiran, for accepting our invitation and to understand the field of bioinformatics, hands-on training with the students, staff, and the research scholars who attended this workshop. And especially my thanks to the 3 colleges who are worked tremendously and made students to understand the importance of bioinformatics. The Government City College, I would like to convey my thanks to the Pushpalata Garu, head of the Department of Biotechnology, Government City College, and Annapurna Garu, head of the Department, Tara College, and Dr. Sudhakar, head of the Department, Acharya Nagarjuna University. At the same time, the other faculty members who helped us, and Dr. Prabha from the Ramaya Institution, Bangalore, and they are worked very well, and they supported us, and they made every student to understand for this workshop. And one more thing, I would like to give this the basic statistics of this workshop. More than 65 participants are registered for this workshop, and among them, the 25 students are enrolled for the basic one, that is the module 1, the basics of bioinformatics, and 50% students are from the MSc graduates, and the 20 to 35% of students are from BSc graduates, and 4 to 5% are faculty, and 10% students are from research scholars. So this is the statistics of the participants who are attending this program. So now I would like to take the pleasure to invite for today's resource person, Dr. Chikoo. Please proceed, ma'am. Thank you. Welcome you, ma'am.
Thank you, Dr. Sambashiva, for the very warm words. I just want to say thank you, everyone, for coming here to this workshop. First of all, I would like to thank you all for extending your support for this workshop, and Dr. Sambashiva for making the initiative to conduct this workshop. So I just want to ask, before proceeding to the workshop, how many of you have a basic understanding of bioinformatics? Can you put in the comment section? Or and how many of you are very new to this topic?
Sorry to Chiran, most of the students they want to learn the basics of bioinformatics, and they want to choose their career in bioinformatics. So the BSc students are having no knowledge on bioinformatics. The MSc students are having a little bit knowledge.
Okay, great.
Yeah,
so I think we can start from the basics. So, yes. So is my screen visible?
Yes. Please presentation mode. Choose the presentation mode.
Okay.
The full screen will be visible. Yes, fine. Thank you.
Thank you so much again. So we are going to the topic bioinformatics basics and databases, and this is the module 1. So in this workshop, you will learn about the different terms and terminologies that are used in bioinformatics, as well as the important databases that are being used in bioinformatics. We all know that now, because as Dr. Sambashiva has said that due to the pandemic, a lot of hindrance has been faced by the life science sector, that you cannot go to the labs. So then this can come in handy, and now a lot of institutions are preferring bioinformatics people all around the world. So this workshop will empower you to learn some topics that is very much related to bioinformatics. So first of all, what is bioinformatics? Bioinformatics is defined as the application of tools of computation and analysis to capture and interpretation of biological data. It is the interdisciplinary field which harnesses computer science, mathematics, physics, and biology. So in simpler terms, it is very easy to say that bioinformatics, to solve problems in biology and medicine, we are using maths or physics problems as well as computer science. So according to Dr. Shashi Shekhar, it is the marriage between computer science and molecular biology. It is in simpler terms. Hope I got your attention now. Okay, now Important terms you will come across in bioinformatics. First of all, DNA and what it is. Now, many of you might be thinking that how I inherited my eye color, my skin color, my height, the genetic diseases, how I got it. Well, it is through DNA or deoxyribonucleic acid. And it is a hereditary material in humans and almost all other organisms. So like this cartoon you can see here, I hope you can see my cursor. So then this is the double helix structure, and this middle one is bases or base pairs. So now the information in DNA is stored as a code and is made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. Now we all love pairing. We love to pair with people we love. So DNA base pairs with each other, adenine pairs with thymine, and cytosine pairs with guanine to form units called base pairs. So in DNA, it is ATCG. Now, apart from this, each base is attached to a sugar molecule. This is a sugar. I hope you can see my picture. So each base is attached to a sugar molecule and a phosphate molecule. So together, the base, sugar, and phosphate is called the nucleotide. I think I'll repeat it again because this is an important thing. So we have got the base, that is adenine, thymine, cytosine, and guanine. So that base, it is attached to a sugar molecule as well as a phosphate molecule. Now this entire unit is called a nucleotide. Now nucleotides are arranged in long strands that form the spiral called a double helix. So this is the DNA and it is made up of nucleotides. Hope this is clear. Any doubts so far? Can unmute. Okay, so we'll go to the next. Next is RNA. Now, what is RNA? Now, DNA is double helical, but RNA is single-stranded. So, ribonucleic acid, RNA's full form is ribonucleic acid, is a molecule similar to DNA. Unlike DNA, RNA is single-stranded. RNA strands has a backbone made of alternating sugar and phosphate group. So, in here, in DNA, it was adenine, thymine, cytosine, guanine. But here, instead of the thymine, it is uracil. So that is one difference between DNA and RNA. And here it is single-stranded and DNA is double helix. So it is a little bit different. DNA, I can say, is found in mostly all eukaryotes, mostly, and RNA is found in prokaryotes, and it depends. So as the organism becomes more complex, so then we can have DNA, but it has both DNA and RNA. Next is protein. A protein is composed of one or more long chains of amino acids. So the sequence of which corresponds to the DNA sequence of the gene that encodes it. So that means you can consider a protein like a chain. So each bead is called the amino acid. So there are different types of amino acids. There is proline, valine, leucine, isoleucine, tryptophan, 20 amino acids are there. And these amino acids are arranged in a sequence to form a protein. Now, this structure that you see, the snake-like structure, is a protein structure. So the individual, it is made up of amino acids. Okay. So then this protein, if it is a single sequence of amino acid, it is primary sequence, primary proteins. Then there is secondary proteins when complex amino acids are involved. Then the structure is called tertiary structure, and the final complex structure is quaternary structures. So we'll have a quick review. DNA. And it's a hereditary material. It consists of four bases: adenine, guanine, cytosine, and thymine. Next, we have RNA. It is single-stranded, and it has adenine, uracil, cytosine, and guanine. And protein, which is made up of amino acids that contributes to the different types of protein that plays a very important role in structural, mechanical, biochemical, and cell signaling. processes in the body. Now there is an entire structure called central dogma. Central dogma is nothing but DNA is transcribed into RNA, and RNA is translated into protein. Let me explain this with an example. Now, if you want to tell the DNA to make a protein, you can see this cartoon. So you can tell the protein, the message goes to the DNA. Hello, I need a protein. So then what it does, RNA, the mRNA and the amino acids start making up the protein. It's like going to a restaurant and ordering a dish. So this is how it happens. So the different processes, this is just for an example I gave. So then the different processes that are involved is that replication. A double-stranded nucleic acid is duplicated to give identical copies. This process perpetuates the genetic information. Next is transcription. Now, DNA is converted into or transcribed into a single-stranded sequence of RNA. Then this RNA, which is present in the nucleus, moves into the cytoplasm. And then translation. That is, the RNA sequence is translated into a sequence of amino acids as the protein is formed. So I hope this is about DNA, RNA, and protein. Any doubts before we go further? Okay, I see no doubts are there. Hope it's clear. Now, the components of bioinformatics. Genomics. Genomics is a branch of science studying structural and functional organization of a genome. So this studies deals with NGS, microarray, then gene-gene interaction studies, gene pathway studies. So this is very important, and it is very much in demand right now in the life science sector. Next is proteomics, or computation chemistry, or cheminformatics. So I specialize in proteomics. So I have a little more depth knowledge in proteomics. So it's the branch of science focusing on proteins and their function and interaction in living organisms. So this we can come across topics like docking studies. Now that is very much in vogue right now, very much in fashion right now, very in demand, because I can say that before you had to do wet lab studies, you have to take a phytochemical or a compound and then see whether it is reacting to your target protein. And it's a very big meticulous process. I think BSc students and MSc students can very much tell me about that process. It's quite tedious. But now with docking, if you have a list of compounds, maybe millions of compounds, you can dock it or bind it to a target protein and see whether it reacts or not. Then you can validate it into your wet lab studies and see the toxicity studies and many other things. So right now, a lot of pharmaceuticals and other CROs are doing this process of docking study. So it comes under proteomics. And this is computational proteomics, not mass spectrometry and 2D gel and all of that. This is for computational proteomics. And the last is metabolomics, is the branch of science studying the particulars of metabolic pathways. Now, I can explain this by telling that, suppose you consume a food. So how that food changes the metabolic pathways of the body is, it's an example I'm telling. It is via metabolic pathways. So how it changes the pathogenesis and the functions of the cells in the body, we can study through metabolomics. So I think I have finished the introduction. Now, I hope everybody is sitting in front of your laptop and you have good internet connection, because now we'll be going directly into the workshop. So is my screen visible now?
Yes, Dr. Chikoo.
Okay, so first database, so you can go to Google Chrome.
The slide is not changing. The slide
a slide. No, I have opened Google now.
No, it's not opening here.
Okay.
Screen is not showing that.
Okay, 1 second. Yeah, now?
No, it's open now.
It's open.
Yes.
Okay. Now, the first database which I want to take you to is GenBank. I hope the students are doing with me because it's like a workshop. So, we can do it together. So GenBank comes under the website of ncbi.nlm.nih.gov. So now we can see that this is a database that was developed after the Human Genome Project, which was discovered in 1990, has been completed. So the different genome sequences has been deposited into the NCBI, and GenBank is a part of it. So we can view the different gene sequences here. So it is very useful, and you can use it for next generation sequencing as well as other applications also. So it's very simple to use. So what we can say is that here we have got a whole lot of options we want. So I'm going with the first one, nucleotide. So I am putting the disease, suppose cancer. Now we are seeing that there is a lot of information. That means about 722, 911, 116 hits are being generated. That means these many hits are being generated. So what we can do is that we are seeing that, see, there is so many of them. So based on the requirement of your study, you can take it. So what is there on the left-hand side? There is based on the animals or the species, there is animals, plants, fungi, protists, bacteria, archaea, viruses. So we can see that there are about, see, in animals, there are these many hits are there. Plants, there are these many hits. So based on that, you can filter also. Then there is molecule types, genomic DNA or RNA. Whether it is a DNA or RNA, mRNA, RNA, you can customize it. Okay. So based on that, you can select it. Then you can see from where it is derived, from INSTC, RefSeq. Then the type of sequence, whether it is a nucleotide. EST is expressed sequence tags. Then GSS is to see whether it is a genomic sequence. Then there is genetic compartments, chloroplast, mitochondrion, where it is present, plasmid, plastid. Then from sequence length, from 100 to 1000. I mean, 100 to 1000, I've just put a random number. You can also put 50 to 1000. It's up to you. The sequence length, that means how long the sequence is. Then the release date, that means when it was released. So I'll go to the first hit and we'll see how it is arranged. I hope my screen is visible. Okay. So what is there? GenBank, there is Cancer Borealis voucher. So then this is the heading. So this is just an example. So then what is that? First of all, there is FASTA and graphics. What is FASTA sequence? Now, let me take you there. Now we studied, I just told you about the base pairs, adenine, thymine, cytosine, and guanine. So you can see that there is different bases here, adenine, thymine, guanine. So this sequence, I can say, this sequence is your DNA sequence. It is very important, and it helps you in understanding how the genes are made, can be made up, and how you can use it to compare with other organisms, like, and how similar it is, or if you have a new sequence, how related it is. Like, suppose you have mice and humans genes. So then based on this sequence, you can compare that by running something called BLAST. Now, FASTA sequence starts with an accession number. This is like a roll number, and then the title, and then where it is present. So I'll show you like how to run BLAST in just in a couple of minutes. Before that, let's see how it is arranged. First is locus. This is your locus, and accession number is the same, just like what I said, your roll number. Then base pairs, 331 base pairs are there. That means that ATC, this one about 331. That means 331 is there. Then whether it is a DNA or RNA, this is a DNA. Then there is whether what shape it is, linear, circular. So then it is a linear one. And this is the definition, Cancer borealis voucher. So this is how it is. So the source is mitochondrial Cancer borealis, Jonah crab. So this is from Jonah crab. It's a crab. And so the organism is Cancer borealis. That is the scientific name. It is derived from Eukaryota, metazoa. Then it's a crustacean. We all know crabs are from the phylogeny Crustacea. Then we have got the references. Then we have got authors who have contributed to this study. Journal, reference, title, journal, comments, to see also which method it was sequenced. So this was sequenced by Sanger dideoxy sequencing. Then source is there. Then rRNA, to see there is a large subunit ribosomal RNA. And then here, this is your sequence, your FASTA sequence. So what it does is that, this ATCG is has 331 base pairs, and it is divided into 6 groups: 1, 2, 3, 4, 5, and 6. So 1, then this is the ladder, 161, 121, 181. This gives the position of the bases: 161, 121, 181, 241, 301. And this last one is 331. So if you want to see the position of the or the locus of the base, you can see it in the sequence. Now let me tell you something. Even a slight change in the bases, A is suppose T, it will give a considerable change in the phenotype or the characteristic of the organism or the functionality of the protein. Yes, I think somebody has raised their hand.
Gayatri?
Yes, ma'am. Yes, sir. Sir, I have a doubt.
Yes, yes.
Sir, ma'am, actually, this genotypic pattern is about cancer, or is it is present in crab?
Okay, this is about it's in crab. So then a good doubt actually, because this one is actually a gene present in crab, because I think when I put crab cancer, I think this one came up. This crab has come out. So I think I will do one thing. We'll go to more gas. So actually that is about the crab genome also, but it is the same thing we are focusing for diseases also. If you want, you can also put organisms here, like Homo sapiens, then many others based on your scientific name, like Mus musculus, then Homo sapiens, Helicobacter pylori, you can put it here. So, okay, so I'll take another example. So, yeah, so this, I just explained what it is. See, so this is the Homo sapiens cancer, okay, gastric cancer associated transcript 3. Okay, so this is the one. So then this is locus, and version is, version is there. This has been updated. So the next version is 0.1. So this is about the title and all of that. And this is your sequence. Now, if you want, we'll now this is for about 961 bases are there. And we'll do one thing. We'll copy this, Control C. And what we'll do is run BLAST. Now, what does BLAST do? BLAST finds similarity between other sequences. I hope you all are doing with me. Can we do it together? So if you want, we can do it together like a workshop. Is that happy? Is that possible, Dr. Sambashiva?
Yes, they'll follow you. Just proceed
Okay.
the things that what you're doing.
Okay. So this is the FASTA sequence. So then we are taking the FASTA sequence. So what we have to do is just take the, no need to take the starting. Now a FASTA sequence, it is followed by a greater than sign, then your accession number, then the Homo sapiens, then to know what RNA or DNA it is. So then what you do is you take the, copy the sequence and run blast. So what you have to do is you have to remove the spaces as well as numbers.
Ma'am, how did you go to the blast?
Okay. Blast, one second. Here on the right hand side, can you see this? Run blast?
Yes, ma'am.
Yeah. So is that, does that, so just click on it.
Okay, ma'am.
Okay. Now there is blast N. There are different types of blast are there. Blast N for nucleotides, blast P for protein, that means like amino acid sequences, blast X, and T blast N for nucleotides and blast X. So then I'll be showing you blast N right now. So I was showing that. So I hope you all are doing with me. So, or I can give you time also to do it, and then you can ask doubts if you have. So you can observe now and then do it also. Remove the spaces and the numbers. It's a little bit of a tedious process, so, but this is very important. Or if this is a little bit of a tedious process, what we can do is we can just enter the accession number. I think all of you are familiar with the accession number, like the roll number, which I just said of the gene, and then rest all will give. Then you can also put align 2 or more sequences if you have one more, if you want to compare with another sequence from another organism or another gene. So right now we're not doing that. Rest all you give what to say, default. Blast it. Message ID mismatch. Okay. Yeah, so your results has come. So that gastric cancer gene, that this one or a non-coding, it has a similarity with 100% similarity with pan-piscus. Then these are all from different organisms. So you can see that there is a total query cover. That means entire coverage 100%, and accession length is 984. So we are seeing the similarities between different organisms in the sequences. So any doubt until now? can I go to the next topic?
Madam, can you explain once again, madam?
Sure, I'll do that. Okay, so what we can do is, see, this is something called FASTA sequence. This is the sequence that consists, it is the DNA sequence. So we've got cytosine, thymine, guanine. So we are going to see how this sequence is similar to another sequence in another organism or another gene. So what we can do, go to the right-hand side and there's something called run BLAST. Click on it. And here the accession number. So then this is a nucleotide. So this is NR minus underscore 126.559.1. So since we don't have another query sequence that we can compare with, so we can compare this sequence with all the other sequences in the GenBank database. All you have to do is press BLAST. That's it. So now this gastric cancer gene has similarity with, see, in different, this is in Homo sapiens. Now there is in different organisms are present. So we can see the different percentage identity similarity is 100%. Accession length is your gene length. Query cover is how much coverage is there, how much is being covered. And this is all about BLAST. Am I clear now?
Chikoo, I would like to add small point here for students. If you allow me from my side. Yeah, students, the BLAST is a searching tool. Suppose example, you have a DNA sample with you. You want to see what is the DNA sample. You don't know whether it is a bacterial sample or viral sample, whatever it may be, you are having a sample with you. When you start the DNA and you submit for the sequencing, you will get the raw sequence with you. So you don't know what is that organism. You have only the DNA. Suppose if you want to know what might be the DNA, whether it is a bacteria, it's a human DNA or some XYZ. So once you get that DNA sequence, just simply blindly dump the sequence in the BLAST tool. It's the completely basic local alignment search tool. That means this data, this tool will generate the data if it is already present in the biological databases, and it will search and it will give the output. Here, if you see this slide, it clearly gives the results of the data that what you have given, and it's shown the similarity. So that means, example, if you take the first one, what madam said now, this is the gene of the Homo sapiens. This is the scientific name of all the organisms. That means the gene that what you are searching is matching with all the organisms, and wherever it is matched, it's shown on the screen. Next, and maximum score. So if you see the total score, maximum score, and query score, and e-value, and the identical accession length, accession number. So each individual gene of the is having their own accession number. They are different. They are not similar. And the position 100% query coverage means the data that what you have submitted in the BLAST tool that is searching and shown the similarity between these data and already existed presented data which is in the databases. So the similarity is 100%. That means the data is matched with all these parameters. And if you see the total scores, it varies. Why? Because the length of the sequence that what you are searching with the BLAST tool, it is matched the positions, exactly how many positions they are going to match in the particular sequence. Suppose example, I found a sequence which is having only 960 base pairs with me, but my data is matching 100%. The sequence which is there in the more than 2000 base pairs of the gene. Okay, it shows the similarity, so that it search and it gives the information that what gene you are isolating and what gene you are studying, and entire thing will be shown on the screen, which is explains about the how to search a tool by running the BLAST. That's it, ma'am. Thank you.
Thank you so much for that point, Dr. Sambashiva. That was very helpful. I think the students got a more clearer picture. So thank you so much. Then we've got pick primers. That is like if you want to pick primers, that also can be. Then highlight sequence features. That means the DNA sequence it will find out, and find in the sequence where the position and all of that we can find out. Next, we are going to so this is all about GenBank, and that is present in NCBI, and we studied about BLAST. Now we are going to something called Protein Data Bank. Now, this is a very important repository for docking studies, as well as studying all about molecular biology, structural biology, computational biology, and others. So how we can find out is that if we want to find out different structural features of a protein, this database is very useful. So like I mentioned before in docking studies, how we can see it using suppose we have a molecule of interest and we have a cancer protein or a COVID protein, and we want to see its binding. We can do using docking tools, but this is the precursor database to do it or to retrieve the data. So let's see COVID protein. So you can see. Yes, so there are 6 structures are there. So this is the database. So we can see in the left-hand side, scientific name of organism. That is, it is in Homo sapien or source of organism or severe acute respiratory syndrome, coronavirus 2. Then taxonomy is eukaryota or ribovira. Then the experimental method, how it was isolated is by electron microscopy. Then to see the polymer entity type, whether it's a protein. Then there is the refinement resolution of the protein. So it is 3 to 3.5. Mostly we prefer proteins which have resolution less than 2 because for a more clearer picture. Their enzyme classification. So we'll do one thing. We'll go to this protein. 6K7. Always remember one thing, that the PDB accession number is an alpha numeric number. So then it is 7. So we've got numbers and letters. So 7KMB. Click on it. Now, so you see this. This is the structure of the protein. So let's go to this page first. So this is the heading. Then we've got, this is the literature, the PubMed. Then we've got, there are different chains are there in a protein. So this is chain A. So there is chain A, B, C, D. It depends on the number of chains. So that is given here. And sequence length is 597. Now, one thing to note here is that, see the FASTA sequence in this one. Now, this is given by first the greater than symbol, then accession number, then the chain of the protein. Then also you see that it is different. There it was a DNA sequence and this is a protein sequence. So you can see that these are the S, T, I, E, Q, A, K. So these are the different amino acids. So please keep in mind, these are the different amino acids. So S stands for serine, threonine, I is isoleucine, then Q is others, and asparagine, and all of that. There are 20 of them are there. So I can say that these are completely different from the DNA sequence, and each of this amino acid corresponds to the functionality of the protein. So now let's look at the, so I think that is, I hope that is clear. Any doubts in it? No. Okay. So what we have got to do is go to the 3D view. So this is a beautiful protein structure. See this, you can rotate it. and see the structure, how beautiful it is. This is the covert protein. Now you don't have to do wet lab studies to get it. This has already been deposited in the PDB database of the covert, recently found out. So then see, now when I click on this thing, see S, can you see that there is, okay, or if I press here, see this, see the sequence. This is arginine. This is glycine. So the different amino acids we can actually find in the protein structure. So it's a very helpful and a useful tool.
Ma'am, can you repeat again? How did you get the sequence of this protein?
Yeah, I'll do that. Go to display files, FASTA sequence. Click on that. So when that comes, you get this page. Show it again. Display files, FASTA sequence. Okay. Or you want it in the picture?
Protein structure too, ma'am.
In the structure. Okay.
Yes,
okay.
ma'am.
This is structure summary. Now there is something called 3D view. See this tab above here. Above here, there are many tabs are there. So then there is structure summary and 3D view. So you see this down. See, you just have to hover over the structure. Or if you want, you can hover over the sequence. It will detect automatically in the structure or vice versa. Am I clear now?
Yes, ma'am.
Great. So next is annotations. Annotations are important. Now, sometimes, you know, there is something called chains. A chain is chain A and chain B. So yes. One second. Okay. The annotation chain A and chain B, it belongs to the family of, there are two chains, and then it belongs to the family of peptidase. These are more of the details of the protein, what we want. So if you want to learn more about the protein, you can go to this tab. Then we've got how it was detected. So it was detected via an electron microscopy, and all of these, this is, I think, wet lab details. So this one you can also go through. Then something important is sequence. So this is the sequence. And genome, where it is present. So now we'll go back to the structure summary. Now for doing the docking studies, we have to download the protein. So what we can do is you can download it in a PDB format. So go to download files, PDB format. Am I clear? Okay. So download it. So you will get it as a PDB. Now this protein data bank file, it can be viewed in PyMold or it can be viewed in JMold. So it depends on the software you are using. But for using the target protein, mostly we always go for PDB. So next module, I'll be teaching you using Autodock and Pyrex how to do docking studies. That is next week. So this is the PDB we use. This is our target protein. So I hope PDB is clear.
Ma'am, I have a doubt.
Yeah.
Ma'am, like for each protein, we are getting different structures, right? So how would we choose out of all those which is good for our work?
Yes. So first of all, you have to go through literature survey, like which protein you are looking for. Then if it is, apart from that, you can go for good resolution. That means the resolution should be low. And which has complete domains resolution should, I'll show you that. So when I was doing my work in 3-bigs, so then what is, see here, the resolution is a little bit big, 3.39 Armstrong. So I go for 2 and below. And so that is one thing. Then you should have good domains and mutation, good domains, that means it should have a complete protein structure. Sometimes it will be a little incomplete. So then that based on the what to say, structure also. And one more thing, do go for recent publication ones. Like I'll show you that. Like look at this. Now if you are going for 2015 2013 or 11, you might get a little more basic structure. So but this PDB is constantly being updated. So then go for the more recent structure. Like this is a good structure. I can say this is 2020 paper. So then based on the recent structures also. Does that answer your question, ma'am?
Yes, ma'am. Thank you, ma'am.
Thank you, ma'am.
Excuse me, ma'am.
Yeah.
Ma'am, what is the AUTHF after chain name, ma'am?
Author.
Oh, author F. Okay.
Yeah. Okay, now I hope PDB, now this is a very important database. So I hope it's clear. Now we'll go to another one. So here we can retrieve all the target proteins. That means if you have cancer protein, then COVID protein, or any other proteins, or, so all of that you can retrieve from this database. Next is PubChem. Now, this is a database where millions and millions of compounds are being deposited. So I can say that I think 3 million or 9 million compounds are there. So what is that? What we can do is that you can also draw a structure. See, you can deposit structures also. See, you can, suppose you want, see, it's very easy. So this one you can do that. Then you can export it also as which format you want. A PDF, image, or a mold file, all of that, we can do that. So what I would like to suggest is, now suppose you have a compound, let's see, aspirin. So you can click on this aspirin. Now we get a lot of details. Now, see, there is a lot of details about aspirin, the structure, starting from the structure, then the here on the right hand side, I hope it's visible, structures are there, 2D structure, then there is the name and identifier, like the IUPAC name, and the IUPAC name is 2-acetyl-oxybenzoic acid, then INCHI, then SMILES, then chemical and physical properties. I can say these properties are important when you want to determine the toxicity of the studies. So like molecular weight, hydrogen bond, donor, acceptor, all of that we can do. Then these are the spectral information. So how it was derived, NMR, all of that. Then related records, like different compounds which is having similarity with aspirin. Then we've got drug and medication, how this drug is useful in treating of the diseases, pharmacology, how it is, whether it has pharmaceutical properties, then use and manufacturing, identification, then safety and hazards, how much toxicity is there, then toxicity is there. So then how much dosage is there, how much we can give, then associated disorders and diseases. So then it has abdominal pain, all of that, acidosis, then literature supporting this document. So then we've got a lot of literature which has used aspirin. So if you have a chemical structure, you can or if you want to search for something, you can put it in the search bar and it will give all these details. Then patents on this drug, biomolecular interactions. Okay. Now this is very interesting. We studied that, we just now studied PDB, right? all of that. And this is one of the PDB structures, like it reacts with, or it binds to 6-MQF. This is the alphanumeric number, and 1 TGM. So then that also it's good. So then if you're writing a paper, so this will really come in handy. Then biological test result, like whether it's active, inactive, all of that. So this is about it. So now for our docking studies, or for our studies, you can download binding studies. So then you can download it as 3D structure, SDF or JSON. Yeah, SDF. So then that is the SDF or mold format. Now, I'll take you, I hope this PubChem is clear. Any doubts? No, okay, good. One second, this one, I'll just move it here. Okay. Next, shall we go for a break? I think it's 11.
Go for a 5 minutes biological break. And
yeah.
And 11:05.
Okay. Okay.
The students, there is a 5 minutes biological break. If anybody wants to drink a coffee, tea, or something, please have it and come back in 5 minutes. Thank you. Yes, we can start.
Okay. Okay. So is my screen visible? Google screen, is it?
Okay. Yes, ma'am.
Okay, great. Thank you. So next we are going to something else called, we covered PDB, PubChem, then GenBank. Now we are going to something called string database. This is a very interesting database, and I really like this database because it gives a lot of information, and without going too much into bioinformatics, also you can have a slight understanding into it. So what you go is, this is the database, type string database in the search bar, press, then you'll get welcome to string. Then these are the different organisms. These many proteins are there, and you can see the interaction, gene interaction, or protein interaction network. So click on search. Now this is very nice. So protein by name or gene by name, you can put your gene by name, protein by sequence. So then if you have your sequence, your DNA or RNA or a FASTA sequence, you can put it here. And you can also put multiple proteins. That means you can put different list of genes, then multiple sequences you can put. And so proteins with different values or fold change or anything of that sort. So let's go into this. I'm going to put protein by name. So there is this breast cancer gene, BRCA1. So I'm going to press. So yeah, so there are different organisms are there. So I'm going to press Homo sapiens. Select, search. Now there is a lot of information that is being told. Now Homo sapiens, BRCA1, breast cancer type gene, and all of that details is being given. So press continue. Now look at this. This is such a beautiful diagram. I can say that this is our query gene, BRCA1. So then we can see that it is linked to other genes. I hope you can see that. The other genes, it is being linked. So you can see the gene-gene interaction network. So these are the other genes that is, it is interacting with. So analysis. Now, this is the detailed analysis. So what it does is that the number of nodes is 11. You can even increase the number of number of genes also. Right now I'm not, so you can press more. See, many genes are there. Okay, many genes are there. 1 second. Just want to, okay. Okay, many genes are there, and you can see that it is BRCA1 is linked to another cancer gene, P53 gene, and other genes are there, RPA1. So the details are all given. So there are number of nodes is 21, number of edges, that is how many, the linkages, all these linked lines, these are the edges. Then we have got the average node degree, then average clustering coefficient. That I'll tell you when we learn cytoscape. So, so the biological processes now, these which all processes, that means which all functions these genes are involved in. So cellular response to actinomycin, negative regulation of helicase activity, cellular response to UVC. Can press more. Look at that. So many pathways like androgen receptor signaling pathways, cellular response to vitamin, all these pathways, it gives how these genes are linked.
Ma'am, what does strength indicate there, ma'am?
Where is that?
Like beside this content network, we have strength.
Yes, how strong this linkage is. Does that answer your question?
Ma'am, like what are the units in which they're measuring the strength?
Yes, yes. I'll show you one thing. So, exports. So, as you can download the details here. So then you can download it as a XML file. Sorry. Or TSV file. What was your question? Sorry, I missed it.
Like, ma'am, in what units the strength is being measured?
Well, it is not in units. These are not units. These are just values. Got it? Like the strength, we'll go to that again. Like score 1.585. There is no like in I think you're in biotechnology, there is like molar and millimolar and all of that. Here it's not like that. It is like scores. It is like values. Is that fine?
Yes, ma'am. Thank you, ma'am.
Okay.
Ma'am, on what basis these values are given?
Or it is the strength. Means how strong the linkages are.
Ma'am, which number will be the highest strength?
It will be this one only. Say suppose out here like 0.996. Can you see that? So there is better interaction between ATM and MDM2. Okay.
That means less number will be the highest strength, no?
No. I can say higher number will be the better strength.
Ma'am, can you repeat this again?
Sure. I'll do that. Okay. Now, this is the string database, and we've got a protein by name. So we are going to put our gene BRCA1, and we are going to click on organism. So a list of organisms will come. I'll just wait for that. Yeah. So Homo sapiens, go down, select, search. Okay, is that clear now? How you got that?
Yes, sir.
Okay, now we are going to Homo sapiens BRCA1. Now we are going to continue. Okay, there you are. So this is our query gene, and these are the genes where it is being linked. So then now we can see that there is different values. here how they are linked. ATM with BAMD, then our gene query. Let's go to the next page. BRCA1. So you can see that, see, look at the linkage. It is a good linkage, 0.999. So I can say that it is a node 1. And it's called nodes. In this context, it's called nodes and edges. So you can also download it as a TSV, or you can download this according to what you want, like a file download.
Ma'am, what does that score mean?
The score means how good how the strength of the interaction is, how well it is strong. Got it? Means like node 1 and node 2, how good it is. I can say that 1 is the maximum score. This is 0.999. 1 is the maximum score. So then this one 0.999, it's a very good score. Is that fine?
Ma'am, a string will basically tell about the interaction between the genes or something else. Okay.
Yes, it will tell about the interaction between the genes.
Ma'am, so the node 1, node 2 when interact, it will lead to breast cancer.
Not exactly. I'll say BRCA1 is a gene. So then this entire, see, the more, I'll show you, more pathways you give. Look at that, more pathways you give. So then this, what you have to do is, go to analysis. See, now what will you have to do is that there is different things are there. Biological process, cellular response to actinomycin. So just see, suppose if it is giving some inflammation, we all know that cancer is inflammation is a cancer, is a characteristic of cancer. So if it comes inflammatory pathway or angiogenesis, so based on that, like ubiquination, so that you have to Google and find out, or you have to go through literature survey and find out whether this pathway leads to cancer, like that. So then I can say, look at this. Now you click on this. Say I press this, protein K6 linked ubiquination. Example I'm just giving. See, these two proteins are involved in this pathway. So whether this pathway is involved in cancer genesis, that we have to find out. So does that answer your question?
How would we find out, ma'am?
Well, that you have to go through literature survey, in-depth literature survey. So then you have to type like protein K6 linked ubiquination related to cancer. On that basis, you have to find out. So many literature articles will come out. So then you can strengthen your paper using this. You do your analysis in this database, and then you have to validate it with literature also.
Oh, we cannot get the information about literature in the string, ma'am.
Well, this is all through this entire thing. I can say is literature, is through literature. But if you want to validate it, so you can see so many processes are there. So let me see. Control F, cancer. Yeah, see, references is also there. You can get, click on this. See, this is the different papers. Can you see it? Reference publication.
Yes, ma'am. Yes, ma'am.
Yeah. So you can get here, but I would suggest go more in depth into the literature survey. So,
Ma'am,
yeah.
What is count in network and false discovery rate?
Yeah, count in network is, yeah, I'll show you. Count in network. See, count in network, it was given 4, right? So 4 genes are involved in that network. 1, 2, 3, 4. And this count is actually done by something called false discovery rate. It is actually a characteristic in differential expression of genes. So that is another topic that is based on significance. Based on significance is how these genes are expressed in the pathways. Got it?
Ma'am, like for that particular option which you choose,
yeah, I'll show that again. I'll show that again. Okay.
No, it is like 4 out of 4, no, ma'am.
Yeah,
for some, it is given as 19 of 10,932.
19 of?
Can you go up, ma'am? Like 12 of 160, like there are more numbers. Those many nodes are not there in that particular network.
I'll show you why it is like that. Now you can actually press more. So you see this. So as you press more, it will come. More genes will come. Hope that answers your question.
Yes, ma'am. Thank you, ma'am.
Okay.
Ma'am, what does PPI enrichment p-value mean?
PPI enrichment is protein-protein interaction. And p-value is significance.
Ma'am, what about the local clustering coefficient?
Can you repeat that?
Ma'am, local clustering coefficient.
Okay. Oh, yeah. Okay. Local clustering coefficient. That is actually like how many genes are being clustered. That means of this clustering means how many genes are being present in a network. So that is called a cluster. So that we can give k equal to 1 till k equal to 15. So that I will, if you are present in the lecture, that I'll explain more in detail in cytoscape. So then that you can find out through a tool called MCode. So that will give how the clustering value, that means how much is being clustered in a particular network.
Ma'am, if it are all the genes which are present in the interaction, ma'am, if only the breast cancer will occur, is it right?
Well, it depends. That's a good question, but these it's not necessary that if all these genes are it leads to the it depends on how it is being expressed, how these genes are expressed. If there is a mutation in these genes, and if there is some problem in the coding of the gene, then breast cancer will occur. But it is not entirely, but yes, you can for research purposes and all, you can definitely rely on this database.
And basically, if comes to cancer, oncogenes which are having mutations will occur cancer now. Ma'am, what is these interactions which are based on the cancer, ma'am?
Can you repeat that question again?
Ma'am, oncogenes are basically occurs cancer in human beings.
Mm-hmm.
If proto-oncogenes have any mutations in their gene sequence, then oncogenes will occur and the cancer will be expressed in the human being.
Mm-hmm.
Then what are these genes which have mutations will cause cancer, ma'am?
Well, see, these are not, it's not necessary that all of these genes should have mutation and all. See, these genes, many genes that are present here, like BRCA1, that on mutation of it, it leads to the cancer. So then this interaction with the other genes, it leads to a pathway. So that pathway also contributes to the pathogenesis of cancer.
Okay, understand. Thank you.
Okay, thank you. So this is about string database. I hope this is clear. Any other doubts?
Ma'am, these genes cause cancer, right? But if they are inactive,
it's not necessary. They'll cause only cancer. They are present in, and like see down, there is a lot of see kick path. There is a lot of see, it leads to p53 signaling pathway that leads to cancer, prostate cancer. So reactome. So then yes, continue.
So all these genes will take part normally in the body, like take part in any pathway for the production of proteins, or will they be inactive?
Well, see, it depends on a trigger. When you see, mostly these things are silent, but sometimes it gets triggered. Once it gets triggered, if there is an abnormal function that is happening, then it will lead to the pathogenesis of the disease. So then you can see here disease gene in association. You can see the Fanconi anemia complementation, Fanconi, all of that. Then there is ovarian, and many other things are there. So breast cancer, see, breast cancer is there. So this all needs a little bit of a tapping into the literature, but I can say that this is a good reliable database to find. But you can also import this string network into Cytoscape and find more characteristics. So then we'll do that in the third module. So we'll go into that. That's a tool. So that is about string. I hope this is clear.
Ma'am, through which type of lab studies these interactions has been predicted, ma'am?
Well, these are actually through, I can say, see, literature survey has been, I can say, references are here. 1 second. Yeah, references are here. See, publication. See, 2021. So then it goes through these things has been validated. Microarray is one. Then NGS also is another one. Does it answer your question?
Those are tools, ma'am?
Yeah, these are tools. And also they have been validated in wet lab. Microarray and NGS, next generation sequencing.
Okay, thank you.
Okay. Okay, now we'll go to the next database. Stitch. So Stitch database is a part of EMBL. So now this is a very interesting database, just like string. Now what it does is that now I'm going to put, let's go to PubChem only and aspirin. Yeah. Now go to the names and identifier in canonical smiles. Now every I can say drug will or a molecule will have a canonical smiles. It is a representation of like carbon, oxygen, all of that. It is CO double bond, all of that. It is being expressed as. So you copy that, put here. Now you put smiles. Now see this. Put this here. This is a little bit similar to a string only. Is this still here? It's fine?
No, ma'am.
Yeah, once more. Okay, go to the PubChem. Go to PubChem. I have typed aspirin. Then go to the right-hand side and go to names and identifiers and go to canonical smiles. Now copy this canonical smiles into the Stitch database search bar. Is that fine? Okay, shall I proceed?
Yes, ma'am.
Okay, now it's the same thing, auto detect, go to the organisms, Homo sapiens, select, search. It's running, so we'll just wait. Okay, so aspirin, it has detected this aspirin and it has matched it to the query structure. And continue. Now look at this. Now this is your aspirin. This is your drug. Now you can see that it is reacting with other genes also. So you can see that, see, it is reacting with, it is having interactions with PTGS1. This is a inflammatory gene, PTGS2. So this is your drug and these are your genes. So then so what I want to convey is that if you have a drug and if you have a set of genes, you can find whether it is having an influence on the genes using this database. Am I clear?
Ma'am, I'm not getting the result.
Yeah, sure. I'll do it again. I'll speak again. So till here, I hope it's clear. Then you press Homo sapiens, press search. And press continue. Now what we can say is that, see, aspirin, yes?
Ma'am, it's showing that the data is not found. Like I have given the item name and organism, Homo sapiens, but I'm not getting the result.
Just press continue.
It's showing that data not found, and when I search the click on OK, it's like coming again back to the item name and organism.
Search by chemical sequence.
One second. I think is your system a little slow? Just press. We'll do it again. Press your smiles. Smiles notation you have to give. Have you given the smiles notation?
Not madam. Mistake happened in item by name. I think we should search in chemical structure.
Chemical. Yes.
Chemical. First, I have also got the mistake by item by name. Then I have searched in chemical structure. Then I got the result.
Oh, okay. It is in chemical structure. I hope. Yeah, it's in chemical structure.
Yes, ma'am.
Yeah. And now is it clear?
Yes, ma'am. I got it.
Okay, great. Okay, now go to search.
This shows that everyone is attending the lecture. Thank you.
Thank you so much. That's really an honor. Okay, now this one is there. Press continue. I just want to say that bioinformatics is very interesting. It's very interesting. If you put your mind, you can, it's a good place to run away from all your problems. So it's a very interesting place. So this is aspirin. That is the drug, that is the smiles notation structure we have inputted. And the genes which it is showing interacting is PTGS1. This is inflammatory genes. This is also a cancer protein. And PTGS2, TP53, ILC, these are all inflammatory genes. So I can say that, see, aspirin has a direct influence on these genes. So let's go to the data. This is also same. So you can see the biological process, see the pathway, biotic, bacterium, biological process. It's more or less a little same. See, pathway. So then microRNA in cancer. So those genes like PTGS2, PTGS1, IL, these are all cancer genes also. So these has influence in these, aspirin has an influence in these genes. Any doubts? Aspirin, yeah, has an influence in these genes.
Ma'am, this stitch is, this interface is nearly similar to the string interface, no, ma'am?
Yes, but the difference is that you can put your drug of interest. Now, if you see, you can put a list of, now I put aspirin, now PubChem. Now I put another drug, like, can somebody suggest a drug of your interest? Amoxicillin, okay.
Paracetamol.
Paracetamol is aspirin. So I think that's a very famous drug now, paracetamol, due to the cold. So this one, amoxicillin. So (2D structure, title, summary. Amoxicillin is 2D structure. Where is the smiles? I think smiles is not showing for this one. Or how about another one, arpizol. Okay. The smiles notation is not available in PubChem, so it's a little tricky here. Okay, so what I want to say is that you can put a list of drugs you like. Okay, we'll go to drug bank in that case.
Ma'am, in Stitch, like only the smile can be used.
Yeah, mostly only the smiles. That is the thing. Yeah, mostly, yeah, smiles. So you can go to drug bank also. So then, you know, can search. So if you have a drug, you can search it in PubChem. Okay, here one Tylenol is there. So I'll try that. So I think getting a smiles is there are other databases also. Chem Spider is there. So there are other database also is there, like Chem Spider is there, Chem Spider, that also is there. This is a drug bank. So if you want to find details about drugs, this is another database, drug bank online. Then, so what I'm trying to convey is that you can put a list of smiles, press on the organism, search. So you can find different, what to say, different drugs are even, you can do one or two more drugs that are interacting in one network and leading, and how it inhibits a disease, all of that. Okay, here itself there is. So there is another things are there. So you just go down, Magnil will take. Continue.
Ma'am, we can do this directly by giving the drug name also, ma'am.
Drug name, no. Smiles, we need to put the smiles. So you see this Magnil, I put
actually we are getting it, ma'am, like when we search, instead of with chemical structure, if we are searching it item by name, then I think we'll be getting it.
Yeah, ma'am, it is getting item name.
Oh, that's really nice. That's something I'm getting. I think they have included it recently. That's really good. I think I got to learn something new also. Let me see. Oh, that's wonderful. So that's great. That's great. Who said that?
Ma'am, myself, Gayatri.
Oh, good, good, good job.
Gayatri, you will get a chocolate.
Thank you, sir.
WhatsApp chocolate only, not red. WhatsApp chocolate will get. Thank you.
Good job, good job. So we can put different, this I think has come recently. So Tylenol is, sorry, do not find a protein or chemical. Tylenol, okay. It's fine, okay. I think so this sequence, this database is clear?
Yes, madam.
Okay, great. Now we'll go to something called, did I take SwissDoc? No, right? No, SwissDoc. Next is SwissDoc. Now, I was telling something about docking, right? I think I was mentioning a lot about docking. So what we have to do is go to this database, SwissDoc, and go to this one, submit docking. And what you can do is you can put your protein's PDB code or a protein name or sequence or URL or upload file. So I think we have downloaded some proteins. So I'm taking this one, 7kmb.pdb. I think everybody's clear about that protein data bank thing, no? All of that, how we retrieved the protein structure via PDB. Any doubts in that?
No, ma'am.
Okay, okay. So just click on that, open. So here all you have to do is put your PDB structure and you have to put your hit. That means your molecule. Put your molecule that is of interest, and then it will do the docking. So it has come. Search here also.
Ma'am, in both target and ligand, we should put the same thing, ma'am.
We have to put see, this is a I can say a mock docking. So this is like see, our target should be PDB and this one should be mol2. So this one you can get it from there is something called autodoc, there is something called padal descriptors where you can convert your from one format to another format. So I have put this, and then you can press on this one, start docking. I'm not putting now because it will take a lot of time. So I'll show you how the result will come like.
Ma'am, from where will we get that mol2 file?
Mol2 file. Well, you can go to PubChem, not PubChem. You can go to PubChem also. Okay. So draw structure. So see, this is a mol file, export as mol file. So you can get it from here also. So protein, so then your docking will be looking like this. It's my script. 1 second, we'll go here. Yes. So can you see this picture? This is how it will look. This is your target, your protein, ligand. That is your drug molecule, which you want to see. See, now if you want to do in wet lab, it's going to be a very tedious process. But here you can do it in a click of a button. And then if this comes and binds to the active site of the protein, then we can say that the ligand is good. So what it does is that it goes and binds to the target protein via hydrogen bonds. So that's what it does. So SwissDoc will give you this result. So SwissDoc is also a very helpful tool if you want to do minimal docking, but I would suggest going for things like Schrodinger, then also.
Ma'am, whether I want to do it by new drug, ma'am, is it possible to know the interaction?
Yes, absolutely. Suppose you have a new drug and you have a phytochemical or a molecule of you have developed. Definitely you can do using docking.
How does the tool knows, ma'am, where does the docking process will occur?
Well, it is due to different algorithms that are embedded in the docking software. So I can say is that there is Monte Carlo simulation, then there is height theories. Every software will have different algorithms. It will which makes the ligand, that means your hit molecule, bind to the target protein. And also due to the functionality of your hit molecule, and also due to the amino acids that are present in the protein. So that all it considers.
At how much percentage of precision the prediction will be happening?
Well, see, there is something called docking score. So then it comes from the docking score is very good if it is somewhere from -5 to -10. So that depends on different softwares, how we are doing it. So that is another session, a big session. So
yes, that is on module 5.
Module 5. So
yes,
that we'll go into that. So that is this is a very valuable tool. So if you want to do suddenly, you come up with a molecule and you find a target and you want to do the sudden and you want to publish a paper, docking is really, really helpful. So we'll get into that slowly.
Okay, thank you.
Okay. Okay. Now, another tool, another database. This is a metabolomics database. It's called the KEG. I'm really happy that it's an interactive session, so then it's not like a one-way traffic. So really happy about that. So now we have got what did I do? Okay, KEG. Now this is Kyoto. It's a Japanese website, Kyoto Encyclopedia of Genes and Genomes. Now, if you want to see the downstream analysis, or you want to see the pathway of like how the different genes are being, like we found out in string, how each gene is interacting, but this is more of detailed. So if I put pathway maps, go to the left-hand side and something called pathway maps. And we press on inflammation. This is a gene, sorry. Sometimes it comes, sometimes it doesn't come. So let's see. So I think there is some problem with this website. Yeah. Okay, so I'll just explain this. Now, what it does is that you can get the information about this website. So KEG is a database for understanding high-level functions and utilities of the biological system, such as cell, organism, and ecosystem. So what it does is that it connects the different genes and proteins via pathways. So I think there is some technical glitch with this website. Okay, there you are. Okay. So you can see this. Like this is like the different I think some BSc students, biochemistry students are there. So then I think you can understand really well. So then this one, starch metabolism and how the different downstream analysis is taking place. Now we click on those numbers. It is a symbol, glucose 1-phosphate. So this is there. Apart from that, I wanted to go to cancer, but it's just not coming. Okay, there it has come. Okay. Okay. Now you can see that these are the pathways involved in cancer. Now see, suppose this is testosterone, that is the male hormone, that one. And then we can see that there are different genes that are being involved here. Look at this pathway. Is it clear? My screen is clear?
Yes.
Okay. So what it said, these are the different pathways in cancer. So what it does is that you can see the different signaling pathways, tissue invasion and metastasis. That means which all genes, metastasis is, I think cancer is being spread to the other organs and it is come in the final stages. So which all genes is being influenced by ROCK, then there is GPCR. All of that we can derive from this, what to say, database. So you can, if you go to the left hand side, see, suppose ERK signaling, see, suddenly it gets highlighted here. Can you see that? Suddenly it gets, you go to the ERK signaling. So which all genes are being involved? TGF, EGF, PDGF, IGF, all of them are being expressed here in this pathway. So I think there were a few questions from students that how these genes are expressed and leading to the pathways. So this is a clear understanding. So you can see that TGF, EGF, PDGF, all of that is leading to the entire detailed pathway is being represented in this database. So I hope this one is clear. So P53, so you can make a, so then now there are, you yourself can make this pathway. So that is, you can, it's systems biology tool. So that is a little bit of a very little complex one, but it is, but once if you do that, you can publish your papers in impact factors of 10 or 11. So it's a very, you will come up with very good papers once you get to know that. So, and deposit here in Keg, because you can see that it's highly detailed. So this one you can use for studying the pathways in detailed manner. Is this clear?
Ma'am, how would we know what is the starting and ending point of the whole pathway?
That you have to look into it. You have to, there is nothing like starting or see from here you start, see testosterone, dihydrotestosterone, other ligands, it goes downstream, keep on from top to bottom, so, or left to right. Or it's subjective. I can say it's highly subjective. You have to suppose, see, you won't need the whole thing. See, these are entire pathways in cancer, but sometimes you can focus on one pathway. See, like ERK signaling or EGF, these are all sub pathways in the entire pathway, or JAK-STAT signaling pathway. Look at that. Now, see, if you want this pathway, now PI3-AKT signaling pathway, JAK-STAT pathway, these are all really important pathways in cancer. So like that, you have to see, look, you'll get sub of it. I clicked on JAK-STAT signaling pathway, so then you can see cytokine interaction. So these all need a lot of insight into databases and also a lot of papers. So then we'll be covering that in the next modules. So Cytoscape gives a good understanding. Then many other databases and tools also can help you interpret this data. But yes, you have to make an effort to study about the genes and pathways in an interesting manner. So I hope this answers your question.
Ma'am, what I'm asking is, for example, take glycolysis, there is a starting molecule and the ending molecule in that pathway. As like that, is there any starting molecule and the ending molecule?
Yes, I would like to say that, see, a pathway can be triggered by something called cytokine interaction network, cytokine hormone GF. Then that clicks on this, it interacts with the receptor and the whole process is being stimulated. And then the end result leads to anti-apoptosis, cell cycle progression. So then a cell death. Apoptosis is cell death. So this is also a characteristic of cancer. So then there are certain triggers. Hormonal is there, cytokine is there, and many are there.
Oh, okay, ma'am. When it comes to a disease or like that something, there are many pathways that will lead to a disease. Is that right, ma'am?
Yes, absolutely. Many pathways are there. It's not
the interlinking between many pathways will lead to the ultimate disease.
Yes, many pathways it can happen. So KEGG can help you identify those pathways.
These pathways are identified through wet lab analysis, huh?
Wet lab, that is gene expression studies, NGS, then microarray, NGS microarray, NGS and microarray. So yes, wet lab also, I can say even RT-PCR.
Okay, ma'am. Okay, thank you.
Okay. So that was about it.
Ma'am, I would like to add a small point regarding what he was asking about these multiple pathways and everything.
Yes, go on.
Actually, it's just like, it's not only in diseased conditions. In general, also, all these pathways are always interlinked, and that is called as something called as crosstalk between the pathways.
Yes.
So if it is in the diseased conditions, it is either a mutation in one of the pathway or overexpression. That is that only under those conditions it will lead into disease, or else also normally all these pathways and everything are interconnected.
Okay, thank you so much for that info, ma'am. I think that was a good insight.
Thank you.
So crosstalk. So I think a new term, crosstalk. I've heard about it, but I'm getting to know new things. I'm thankful for that. Thank you so much, ma'am, for that information. Thank you. Okay, so I think KEGG is clear. Now, yeah, I think I have covered the major databases. So I think now we can go for an interactive session, or what would Dr. Sambashiva like to suggest?
Yes. Ma'am, is this KEGG will give, I mean, suppose if you have one molecule, some intermediate of some pathway, can you trace out the pathway for that molecule? Like, is it involved in different pathways or any interlinked pathways. Can we trace out those pathways in this KEGG?
Yeah, actually it can give. So like BRCA1, so it is interlinked to pancreatic cancer, ovarian cancer, breast cancer, Fanconi anemia, all of that. It will give. Does that answer your question? Hello.
Sunena
Yes,
madam.
Yes, ma'am.
Yes. I answered your question. Did you get it?
Yeah, ma'am.
Okay. I think I have completed the session. So any doubts or anything you would like to know? So
yeah, thank you. Just a minute. Yeah, thank you very much, Dr. Chikoo, for your wonderful session and workshop, hands-on training. Now the session is open for question and answers. Students, you can shoot the questions as many as you can. If you have any doubts regarding bioinformatics career, basics of bioinformatics and everything, because in today's talk, she has covered all the basics databases, the data, how many databases are there. And the exclusive talks on the proteomics, genomics, systems biology, document studies will be discussed in the upcoming modules. So if I request all the students to discuss about the today's module 1 talk and the concepts, whatever she delivered during the class since 2 hours. So discuss on those things and try to get the information and ask your questions. Thank you. Now the session is open for question and answers. Thank you.
Thank you, Dr. Sambhashiva. So if any questions are there, definitely I would love to answer.
Ma'am, this KEGG will give only, only gene interactions or else many chemical interactions also?
Chemical interactions also it will give.
Okay, thank you, ma'am.
Yeah.
Ma'am, like, is there any difference between the stitch and string database, ma'am?
Well, in string, you cannot see a drug gene interaction, but in stitch, you can see.
Okay, ma'am. Thank you, ma'am.
Okay.